All right, welcome back. In this video lecture, I'm going to cover the second half of A3, which deals with transport inside of cells. And so part one, we looked at passive transport, which is the movement of molecules or water from high concentrations to low concentrations without using any energy. But in this video lecture, I'm going to talk about active transport, which means that we are going to use energy because we are going against the concentration gradient. And basically what that means is we are going from low concentrations to high concentrations. So I like to think of it as like a hill. Okay, when you are at the top of a hill, okay, up high, and you go sledding, you just need a shove, and then gravity kind of takes over. You're not really using energy as you are traveling down that hill. But now we're at the bottom of the hill, okay, and we want to continue to go sledding. So we're at the bottom of the hill, and we have to trudge our way back up, and so we're going to use some energy. And so all of these examples are movement from low concentrations to high concentrations, where we're going against the concentration gradient and we need energy. So one example of active transport is called molecular transport, which means we're going to use some protein pumps. And lots of examples for protein pumps that go from low concentrations to high concentrations: calcium pumps, potassium, and sodium. But in all of these diagrams, you can see that I am moving from a low concentration, and there's my energy, ATP, to a high concentration. Okay, I'm going against the concentration gradient. In my sodium potassium pump, I am going from low concentration. I'm going to transport 3 sodiums out and 2 potassiums in, but I'm using energy. That's the thing here. I'm using energy to do this movement. So, molecular transport, protein pumps. We can also move things in bulk, and there's 2 ways to do that, and that is endocytosis and exocytosis. So endocytosis means that materials are going to be engulfed in a membrane and brought into the cell. And so you might have like receptors on the cell membrane that catch the target molecule, and then it will kind of evaginate or kind of fold in and form this vesicle. And now we've brought substances inside the cell. And so we are bringing it into the cell. This is endocytosis. Now, if it's a solid particle, it's actually called phagocytosis. But if the cell is bringing in like dissolved ions or something, then we call it pinocytosis. So you can see that this membrane makes a pocket around the material, and then it breaks inside the cell and it forms this vesicle. White blood cells do this all the time when they are going to eat bacteria. The other way is exo, going in the opposite direction. We're going to release substances out of the cell, and how it happens is that we have our vesicle, and then it fuses, because they're both made up of phospholipids, it fuses with the plasma membrane to release the contents. Neurotransmitters from neuron to neuron, this is how they do that. Here you can see that the neurotransmitters are packaged in a vesicle, and then it will fuse with the plasma membrane and travel across this gap, where they will bind to receptors and then continue on the signal, so neurons can play a giant game of telephone inside of you. So this is exocytosis. It's out of the cell, exiting, leaving the cell. Don't get it confused with endocytosis, but all 3 of these, protein pumps, okay, endocytosis, exocytosis, are considered forms of active transport. Okay, so in summary, okay, active transport requires energy because you are going against the concentration gradient. We are going from low concentrations to high concentrations. Okay. Passive transport, no energy, okay? And we are moving from high concentrations to low concentrations, either through diffusion, facilitated diffusion, or if it's water, osmosis, okay? And that does it for 8.3, cell transport.